parties or something.
Oh, awesome.
Yeah, so this is the store.
Wow.
But
that looks amazing.
Let's see. They have strawberries.
You know, that's one of my son and I's favorite things to do is to get a little treat at the mall and sit on a bench and people watch.
Well, they
really
have
weird.
ice cream. They have ice cream. They hand spin their own cotton candy.
Hand spin. Wow.
They got cake pops.
Oh,
so
yeah,
if
he's gonna
y'all
like that.
Yep. And
Hey,
candy
Ross?
apples, caramel.
Ooh, I love a candy apple. All right, let's see. Try doing your best, right? Okay, sharing my screen.
And he had 3 orders. He just made the face.
Oh yeah, and I'm supposed to pull up my chat on the other one. I forgot.
And he had 3 orders last night.
That is awesome. Okay, how we doing? And I'm gonna put the chat on here until I can get it figured out. Okay.
So he's been looking for one, hasn't been able to find one. It's really hard. So yeah, so the shelter closed, it's called that schedule appointment. So you have to put a hold on the dog if someone has it before you. I saw the saddest thing.
How you doing, Ross?
The other day.
I'm doing fine.
Yeah?
Saw the saddest
Overworked, overstuffed.
I'm like, oh.
Overworked. Yeah, that's fine.
Just
Because you know, college kids, you know, doesn't give them, you know, college and college professors don't really care too much about everyone, about your schedule or how much time you actually can help just one person. So you.
it is definitely a balancing act, and learning how to make that balancing act happen is huge. Huge. Okay, I think I think we didn't do this one, so we'll leave it on that. That was the last one that we didn't do, right? Okay, and then I need an attendance.
But it's so hard to adopt a dog. It's really cheap to adopt dogs here, though. Like, yeah, but they want you to like do records and all this and that. Yeah, but okay, that's because I know they want them to go to like a really good. No, the thing is like when we used to get plants a lot, people would adopt dogs from the shelter and then either breed them, find them, and use them as big dogs. And then that's how a lot of that happens.
All right.
Bringing your dog's not going to hang you. Just great. My little one is so fun. People don't want to neuter. Somebody's like, are you going to bring your nose?
They don't want to neuter or spay their cats?
They don't want to neuter or spay any of their pets. They're like, it's my baby. I don't want to do that to it.
It's good for them.
Oh no, I spayed mine.
It's good for them.
That's
right.
number one. You lower their
Spay and neuter your pets.
cancer.
Exactly. You lower the chance of cancer. You don't have to pay.
You fix a lot of behavioral problems.
You do.
Mine is
And a lot of like destructive behavior in your home.
Mine, like, if you have a male cat, it's gonna stop.
Okay, let me do this, and let me do. Okay, there's attendance. Attendance is ready to go. And Mason, I don't know what that is. Okay, let's see. I'm gonna change this.
Don't wake up from anesthesia. No, like the next, like you just start.
Oh, I need to open the other one.
Like days after.
So many different things to open. Oh, yes, infection of the uterus. Yes, yes, yes. See, always good to spay and neuter your pets. We're having this big discussion in the classroom about why it's so important to spay and neuter your pets. You never know what we're going to talk about in here.
Hey, it's science.
Yeah, we spay and neuter to prevent many things. This is one of those things. They're participating in our conversations. See, we have some support online too. I love it. Okay. I guess we could talk about many, many things. All right. What else? Oh, I wanted to do a Zoom on here. It's too many things. No internet. It's like the internet does not like my laptop over here.
No, it doesn't like mine either. Every time I come in, it likes mine.
I need to know what the special deal is. Okay.
Are we doing eye-clickers today?
Yes, you're doing eye clickers today.
I just need to know what kind of notebook I needed to get out.
Yeah, we're gonna do we're gonna finish up the long questions from the end of 5.
The Eliza one.
Well, we had a few. We have a few. Eliza's one of them.
Yeah, Eliza.
We have a few. And then we have some short ones from chapter 6. And you know what? I meant to grab like my extra papers in case people wanted them, but I didn't. Everybody got one in here?
I just have that one as my last one.
Yeah. You have that. Wait, you have that one as your last one? There's one more.
No, we got.
And you got the extra? Yeah, look. Do you have that one?
Let me find it. It's section 41. Oh, I have it. I think
Did I really not grab it?
here Wednesday. And
Oh.
I got it Wednesday.
Let me see. I have a stack on my desk. It's so terrible. But they're on Moodle. They're all on Moodle. I wasn't anticipating anybody coming today. Okay. I think we're ready to get rolling, right? All right, before we start, any questions? Okay. I think we will rock and roll. All right, so we still need to finish the last one. So, all right. So the last one, I know this is like from a long time ago, but our solid phase synthesis process, right? So this one was just reordering your solid phase. Oh, and you did do attendance, right? Everybody checked in on attendance? I think a few people need a minute to check in on attendance. So take a second. Okay, so when you enter this, right, there are 5 steps to generate our peptide. So what I want you to do in iClicker is when you enter this, what you're gonna write is like, ooh, if I could write, A, comma, B, comma, C, comma, D, right? That's how I want you to put in your answer, okay? So you should already have the answer to this. I'm gonna stop attendance and I'm gonna make this a short answer. Go for it. I'm gonna go close the door. Be right back. Y'all should already have the answers to these. Yay! What I want is like to see who clicks in. Yeah. That's what I was hoping for these long ones. Like there are some short ones. So we're discussing in class what's better, having the long questions to take home or having them in class. And kind of the consensus is that it's been nice to have them to take home and to think about them.
It's just, I just don't have enough time. I just don't have enough time to review them and study them. I'm too busy doing 5 other things for like 5 other classes.
Yeah. All right, so I'm gonna call it at 2 minutes. You should have your answers in by 2 minutes. Because we had 46 people check in, I think. Got 33? Come on, come on. And I don't have a joke today because my son is with my mom because they closed school today to clean because they're all going back on Monday. So I have no joke. I'm so sorry.
How does a cow do math?
I don't know, how does a cow do math? with a calculator. That's great. That's great. Did I tell you all the chicken one? Oh, then I do have one. I have one. Okay. How does a hen count her eggs? Oh, what do you call a hen who counts her eggs? I did tell y'all this one?
Yes, the
Oh, a mathemachicken.
Oh, yeah.
Okay. So y'all did do that one.
Dad jokes are always fun. So,
Bad jokes are always fun. They are.
both of us putting the wheel on our gate.
Come on, y'all. I can't believe we only have 37. All right. So, what did you say? There are 5 steps to generating our peptide. So the first thing that we have to do whenever we're generating a peptide, we have to remember that we're building this peptide backwards, right? So normally in the cell, right, this is the end. This is the beginning, right? So here's our N-terminus, and we're going to add the next amino acid right here on the carboxy terminus end, right? That's what happens in the cell. But is that what happens when we synthesize peptides in the lab? No. We're going to add to the nitrogen here. So we need to make sure that we're only adding at the times that we want to add. So what we have to do is we have to block the N-terminus unless we're ready to add. So we start out, we have 1 amino acid hooked to our resin, and we have our FMOC attached to it, right? Okay, so what's the first step that we are going to do? I'm going to erase, right? What is our first step? Let's see what y'all said. Okay, so A, C, D, E, B. There we go. A, C, D, E, B. That's also good. A, C, D, E, B. A, C, D. No, that's B, E. It's E, B. No. Oh, did I miss one? A, C, D, no. No, okay, so those were the good ones. If I missed one, y'all tell me. All right, so the first thing that we're gonna do is we're gonna deblock the amino acid that's attached to the resin. So the first step is to get rid of FMOC, because if you don't get rid of it, the rest of it's not gonna happen, right? Okay, now what's our next step? So let me write that down. So we go, here's our order, A. Our next step, we need to activate another amino acid residue that is already FMOC blocked. That's what we want. We're going to add a residue, right? So we need to prime that new residue. We're going to keep it FMOC blocked on the N-terminus, and we're going to prime it. What do we prime it with? DCC, right? So the DCC is going to allow for interaction with the N-terminus, right? And when this primed amino acid, right, comes into contact with our unblocked amino acid on the resin, we're able to make a new peptide bond. And when we make that new peptide bond, we release the DCC, so we don't have a weird modified amino acid. Okay, so that one was, we did A, activation of our blocked residue, okay, AC. Now, we want to couple our amino acids, right? So we prime it, and then we're going to couple our amino acids in step number 3, so that's D. And then what we want to do is we want to unblock all of our residues, because if we don't unblock them, what's going to happen? What if I just stopped here, cut it off the resin, and said, oh, it's good? Well, we're not trying to sequence it. We're trying to make a protein here. We're trying to make a peptide. So sometimes we want to make an artificial peptide because, let's say, oh, we have a protein that we want to crystallize, right? And we know that there are 2 conformations of that peptide. One of them is the bound state, like a receptor, and one is the unbound state. So I know that this receptor binds to this target peptide. So I'm going to synthesize this target peptide using our solid phase synthesis, right? Now, how do I know that that amino acid peptide that I generate is going to be good? If I stop at step number 4, what's the problem? It's blocked. What's blocked? What about all these things? What are these things? that came off. Those are blocking agents for the side chains, right? Because we need protection on the side chain so that you don't add things weirdly. We only want to make peptide bonds. So if we block the side chains to prevent interactions from occurring, that's great for while we're synthesizing. But what happens when we're done synthesizing?
Now remove them.
We got to remove them so that the protein will
Be able to synthesize other.
IEN, protein, I always get crazy. EIN, no, I don't know. Will what? Fold. What allows a protein to fold? All of the interactions between the side chains, right? So if you block all those side chains, is your protein peptide going to fold properly? No, and if it doesn't fold properly, is it going to interact with your target protein properly? No. Okay, so then 4 was E deblocking, right? E deblocking, and then cleavage from the resin and deprotection. There we go. Okay, so now we have our free N terminus, a free C terminus, and all of our side chains are unblocked. So that's what we want, a real peptide. Questions? You do have to understand why we do these steps in protein synthesis. I think that's really important. Okay, let's try an experiment. Y'all should have read this and you should have an answer. So I'm giving you 1 minute to put in your answer, 1 minute only, right? Okay, so we have this experiment. You found 2 proteins, protein X and protein Z. Now, the problem is they're similar size and they have high sequence homology. There are 2 domains of the protein that are nearly identical, but each possesses a different sequence in another domain. What antibody is going to be our best selection for us to identify the expression of protein Z, but not protein X, using a Western blot?
I think you left it on like poll.
Oh, I left it on poll. Oh, sorry, y'all. I'm going to start again. So sorry. Okay, try again. That should be better. Thank you. No, I just was saying thank you. So everybody check in. You got 1 minute. Monoclonal will only recognize 1 epitope. That's exactly right. What? Oh. Well, okay, so a common epitope is like 2 proteins. Even though they're 2 different proteins, the epitope is the same.
Oh, so there's only 2.
So in A is referring to a monoclonal antibody that's raised against common epitopes of both proteins. So A is saying that I have this one monoclonal antibody that'll recognize an epitope on protein X, and it will recognize the same epitope on protein Z.
So it's the same epitope.
Yeah. So, okay, so what you should have looked at this and said, if I'm trying to separate out one protein from another, you should have automatically thought monoclonal antibody, right? So we were able to look at these and just X them out, right? So now the question is A or C. And so in the way that you answer that is to understand what's being asked in the what's being proposed in the answer choice, right? So A, monoclonal antibodies raised against common epitopes of both proteins. That means protein X and protein Y, Z, what am I on? Z. They both have a similar domain, and so this one antibody will recognize both protein X and protein Z. So is that what we want? Do we want a monoclonal antibody that will recognize both proteins?
Yeah. We want, don't we want to like, don't we want to be able to identify both? No,
Wait,
We want Z, but not X.
so we're like, just an unique donor.
So A is not going to cut it because it's going to recognize both. If we look at C, monoclonal antibodies generated against the unique domain. So that's saying that protein X and protein Z have portions of their molecule that are different. And so this monoclonal antibody will recognize one protein and not the other.
So
And-
it's not specific.
So it's what?
So it's not specifically the epitope for, like, to differentiate. Like, it's the protein itself that's, like, say that they're together, like, one's unique and the other one,
Uh-huh.
but they have common epitopes. It's not always like the epitope that is.
The epitope, so wait, so the definition of an
I guess
epitope.
I know what an epitope is,
Okay.
but my question is, like, to express one protein but not the other protein, like, the epitope is not the main factor in to express it. It's
You're not
just-
expressing the protein.
Right, like,
You're recognizing the protein. So the antibody that you make, right? How do you make these antibodies? You
you get sick or they're.
well, you expose, you have an animal, right? You have an animal and you expose the animal to protein X, right? And that animal will make antibodies to protein X, right?
How we get a flu shot.
Right. So either you can harvest the antibodies, which will give you polyclonal antibodies, or you can recognize the cells that produce the antibodies and separate them out one at a time, and you'll get only one, and that'll be monoclonal. So each cell only makes one antibody. So what we're saying in C is this one antibody will recognize protein Z, but won't recognize protein X,
And why?
because the two proteins are not 100% homologous,
Okay, so it's the protein itself, not the epitope to.
but the protein itself, its configuration determines the epitope. So epitopes are locations on the surface of the protein
Right, for the answer.
that allow the antibody to bind to it.
Right.
Yes. So the surface of protein X is slightly different than the surface of protein Z, and this antibody will discriminate between the two.
But the epitope is the same.
No,
Okay. I
no,
think I'm just getting from A to C
yeah.
different.
So we're saying in the problem that the two proteins are very, very similar in shape, and so there are antibodies that will recognize both, and then there are differences in X and Z. And so the monoclonal antibodies, you can pick some that will recognize both, or you can pick a monoclonal antibody that will only recognize one.
So it's not depending on the epitope.
It does
It's
not,
depending on the structure of the protein.
it depends on the structure of the protein, which determines the epitope. The structure of the protein determines the epitope. Okay. Anybody online got questions on that one? You okay? Y'all super quiet today. Yeah, okay. All right. So you do need to know how the different antibodies are made, polyclonal versus monoclonal. That was one of the things that I talked about in the videos, right? How they're produced and what they can recognize. All right, so now here's a practical application of our antibodies, right? And I'm going to talk, but I'm going to give you a minute to enter your answer choice because you should already have an answer choice. So a lot of people are allergic to different things, right? And so one of the things that we try to do is figure out what's causing it. So if you have an immune reaction, you are going to produce immunoglobulin E, right? And that's what's going to trigger our immune response. So if we know what allergen, we can test the food and say, is this allergen in the food? Okay, you shouldn't eat that. But it's a little more complicated when we want to know which allergen, we don't know which allergen triggered the person's response. So how do we figure out which allergen is causing the production of immunoglobulin E? So we have a couple of different ways. So we're trying to differentiate which allergen is inducing the effect, right? So we could use an ELISA. So it's in this case, they're saying a sandwich ELISA to capture antibodies produced against the allergen. We could use mass spec to measure increases in histamine to show if we've been exposed or not. We could use mass spec to determine the presence of antibodies after exposure by the mass of immunoglobulin E, or we could do Western blot. What can we eliminate right off the bat? Which ones? You didn't correct the last question. Oh, I'll go back and do it, Julie. I'll go back and do it in just a second. Thank you. Okay, so what can we eliminate right off the bat? Okay. Why can we eliminate the Western blot? You're right. Why can we eliminate the Western blot? Well, so we're saying we're going to run our sample on an SDS page gel. So if you want to know if somebody's producing antibodies, you're going to take serum. What's going to be in the person's serum? You're going to have, sure, you're going to have IgE. What else are you going to have? IgM, it's actually IG. It's a little g. I don't know why I did big G. What else? IgA, IgG, right? You got all this stuff going on. Can you separate, based on size with an SDS page gel, the different antibodies? No, they're all antibodies. They're all the same length. They have a little bit of variations, yes, but not a lot. So you're not going to have a size difference. So you can't separate them on a gel. So that's not going to work. They're all the same size. What else is not going to work? I always like to look at problems and see what's a big no and X them out first. So how about using mass spec to measure the increase in histamines to show whether the antigen has been exposed to the patient? Does that tell us which antigen the person's been exposed to? No, it's just telling us you've been exposed, you've had a reaction. Congratulations, we already knew that. Answer choice B does not give us any information that we didn't already have. Maybe if we were trying to find out if they had an allergic reaction, but we're not. We already know there's an allergic reaction. Okay, so how about mass spec to determine the presence of antibodies in a person after exposure by the mass of IgE? What's the problem? What's in this one that was in D?
Because we're trying to use the fine immunoglobulin, like we're trying to mass them, and it's hard.
It's impossible. It's impossible to separate them on mass. You're not going to do it. Okay. Wow. So what's left? A, we have to do an ELISA, right? Okay, good. So most. Oh, I got to stop the poll. Ha ha ha. That would be great. Okay, so ELISA. So how does an ELISA, a sandwich ELISA work? Somebody tell me, what's the first thing we're going to do in a sandwich ELISA? So an ELISA we usually do in like wells, right? We have lots and lots of wells. Right. And they have different wells, but the point is that you can do a lot all at once, right? So what do we put at the bottom of the well?
Different ant.
If it's a, think about this, when it says sandwich, right? You think two pieces of bread on a sandwich.
You place one material on top, one material on the bottom, and place something in.
But this section, we're specifically talking about what kind of protein. That's my bread. The bread of this section is antibodies. Antibodies. So we have an antibody at the bottom, right? It's my little Y-shaped antibodies. Why did we put those antibodies at the bottom of the wells? Well, we're actually, in this case, we want to know which allergen the person is allergic to, right? So if we put allergen, maybe this is allergen A, this is allergen B, this is allergen C, this is allergen D, so on and so on and so on, right? We could test a bunch of different allergens all at once. Then what are we gonna pour on top of our, well, yeah, but where'd they come from? More antibodies is right. They come from the patient's serum. So the patient serum after they've been exposed, right? And so you put it in all of them, right? But we don't know which one is going to recognize the allergen. So how do we find that out? Well, let's pretend we have one that did. Let's just kind of zoom in on one. We have one where we had an antibody, and let's say that this was antigen D, and the patient's antibody is right here, and it recognizes antigen D. I know that this patient is making an IgE molecule that recognizes my antigen, right? Now what do I need? But I'm not done. It's almost like a Big Mac, you know? It's not like a regular sandwich that only has 2 slices of bread. This is like a Big Mac that's got 3 slices of bread. So what are we equating the bread to? Antibodies. So that antibody that we just put, we can't really detect it, right? We don't know that it's there. So how are we going to detect the well that has my antibody antigen interaction occurring? I need a Big Mac, I need another slice of bread. So I'm going to do a secondary antibody. So this is a secondary antibody that has enzymatic capabilities attached to it that can work on a substrate. So that's S plus a substrate. And this enzyme plus our substrate will give us either a color change, or there are a couple of different ways that you could do it, but it will give you a visual output.
I was gonna say, kind of lost. I'm not sure how this will work. I'm getting like basically the scent is most effective because like there are multiple like so that's and two because since they're put together, you can test which antibody are reacting to which one.
Yep, so this will tell you, this will give you a reaction, right? Well D will have this color change reaction, and all the rest will be blank, basically. So we'll know that in this well, I put antigen D. So this is what my patient is allergic to. My patient is allergic to agent D because we were able to recognize it with the patient serum antibody.
How does that work? Like, do you just put like a sample, like some sort of some grass or peanuts?
Yeah, yes.
Like, how does that work?
You actually have to sort of purify out the antigen that you want. Like if you are trying to test for somebody being allergic to peanuts or something like that, you need to isolate a specific peptide that typically causes an allergic reaction in everybody else, and that's what we test. So it's pretty specific. Oh yeah, they mass produce them. They absolutely mass produce them. Let me see. Yeah, you dander. Yeah, you can put little pet dander peptides in there. You can, and there are some skin tests too that they do where they inject a little bit like right under the skin, or they put like a patch.
Yeah, I know. I had to go take one of those.
And then they put it on.
I heard I had to go take one of those. They injected like 7 different little.
Yeah.
things into
Okay.
my arm, and
Last one on chapter 5. Last one on chapter 5. Y'all doing okay on Zoom? Like y'all are so quiet on Zoom. All right, so let's say we have a protein and we have a really hard time visualizing it or purifying it. So what do? We're going to add a highly antigenic peptide called an epitope tag to either the amino end or the carboxy terminus end, right? And so it's going to enhance our ability to recognize it with antibodies. So what describes how we produce these epitope tagged proteins? Thank you for the response. And I still have no internet on my laptop. It's a good thing I don't depend on it.
Like it says lost like internet connection, like no.
Yeah, it has like the exclamation point, like.
Mine does that, and I have to go and hook up to a different network. There's like the NICU 2.0 or the NICU 2.4.
Yeah.
I just have to change whichever one.
Did you try like the unwired 2.4?
Let's see, I tried unwired 2.4. That's what I just tried. So I'm gonna try the other one now. But anyway, it's fine. We'll survive. Okay, so should have clicked in by now. I'm gonna do attendance like in the middle of class. I'm not gonna do attendance first anymore.
Still do it, but it's a fake one. Lure people in.
All right, let's see. All right, so A sequence of amino acids common to most proteins in the cells is added to the gene for the protein of interest. Why not? Okay.
That only lights up when it's in the presence of it.
Okay. How about B? A unique peptide sequence like the flag epitope is chemically cross-linked to the mature protein of interest. A lot of times what you're trying to do is you're growing these cells, right? And you want your protein of interest to do its normal thing. You want it to be in the zone, interacting with its interacting partners, doing its thing. And then you want to pull it out and you want to see, you know, is it upregulated when this happens? Is it associating with something else? You want to answer a question about it. So you have to try to get that protein in as natively as possible. So if we have a unique peptide sequence like the flag epitope, that's true. Can we chemically cross-link it to the mature protein? You can do this in a test tube, in vitro. Can you do it in vivo? There's a lot going on in the cell. You cannot do that in vitro. So we don't want that, right? We don't want that. All right. How about an epitope tag consisting of a long stretch of non-polar amino acids is incorporated into the protein of interest? Can we incorporate it into a protein? No, we can't. We can't incorporate it into a protein that's already there. That's not how we're getting the cart in front of the horse, right? We want to make a protein that has this tag on it. How do we do that? You got to put it before what? That's exactly right. You got to put it before what? Before the gene is transcribed, you want it to be changed, right? So what if we take the gene that encodes our protein of interest and we modify that gene? Like we modified our plasmid, right? You can actually modify the DNA. So if we modify the DNA, now the coding sequence will say, code for my protein, code for my protein. And instead of stopping in between the stop codon and the rest of the protein, it'll say flag tag, end. Or at the beginning, you can have the tag. Either way, it can either be at the beginning or the end. But you're going to modify the gene so that when the cell goes through its normal transcription, translation process, that tag's already there. Does that make sense? Okay, so how is A, so we can't do C, how is A different than D? We're talking about the gene here. We're talking about the gene here. So in A, you're adding amino acids to the gene. Does that work? Absolutely not. You cannot add amino acids to a gene.
That's, you can't alter it. They could do that. If you could do that, like
yes, Tristan.
So many. If that was possible, then so many genetic diseases would be.
Yes.
Would be
But
done.
Tristan, yes, you are also right. You are also right that we do like them to be polar. Why do we like them to be polar? So we do want them to be polar. That's a great point, Tristan. Why do we want them to be polar? We don't want non-polar, we want polar. Polar tags. Why do we want polar tags? We don't want to mess with the hydrophobic folding properties. So we don't want to mess with folding. And what's the whole point of the tag? That's half of it, Tristan. That's right.
The whole point of the tag is to help us identify the protein of interest.
So if my protein of interest looks sort of like this, okay, it's my beautiful protein drawing, and I'm telling you that here's the N terminus, and I have my tag right there. That's perfect, right? Now, what if that end is non-polar? It'll be inside. Now, what do I want to use to recognize my tag? An antibody. An antibody that is specific to the tag. If my little tag is folded into the inside of the protein, does the antibody have anything to recognize? Nope, has nothing to recognize. So it's not going to work. My experiment's going to crash and burn. We've all had those.
Because so very D is derived because it needs to be on the outside.
So C is incorrect for two reasons, right? One, because you can't incorporate into the protein, and two, because we want our tag to be polar. We want our tags to be polar, so two reasons. So they don't disturb, we'll say, the quaternary structure, right? We don't want to disturb the structure. So it'll kind of be on the outside, so that'll help that it doesn't go inside the molecule, so it doesn't disturb folding that way. And then the other thing is, tertiary will say folding, right? And the other thing is, we want the tag on the surface. Why do we want the tag on the surface? So the tag can what? Be recognized by the antibody. That was a great side trip, Tristan. It's fun and educational. Okay. Questions on that? Okay. Did I correct that one? Oh. No, oh no, this was my previous one that I had to go back and correct. Here we go. So I did not correct this one yet. Oh, we had a big range on that one. Yeah. Got to be careful when you're reading those options. Okay.
Is it wrong to say that the atoms being bonded by the heat of it's binding.
You could say it's an interaction. Yeah, you can say binding. You can say interaction. Yeah. Okay. I think that's it, right? Okay. Yay, y'all did great. Okay, so moving on to chapter 6. The beginning of chapter 6 is a nice little introduction on all the different kinds of, oh, that is so not the right thing, on all the different kinds of types of proteins and their general functions inside of the cell. I thought it was a really nice introduction. Okay. And how are we doing on chat? We're doing good. Okay. Trying to keep an eye on that chat. Here we go. All right. So remember the book put these proteins into functional classes, right? So we're characterizing the proteins based on what they do inside of the cell. So which one of the following was not one of the major functional classes of proteins? So this is a great big not a functional class. So these are ones that you do not have yet. These are the quick and easy ones. The rest of the day is quick and easy. Hmm? I'll give y'all a minute and a half. And mark off as we go. So I remember where we ended. I'm bad about that. Y'all help me though. Y'all are so sweet. Okay, you got 5 seconds. How many did I thought we had 46. Okay. So what do we say? Good, B, good. Yay! There are no quaternary resistance proteins. That is not a functional class of proteins. We do have transport, we do have structural, we do have metabolic, and we do have genomic caretaker proteins. I like how they say that, caretaker. Oh, well. All right, let's see. How do I zoom out just a little bit? Because I want the whole thing in the window so that it's on your clicker question. Okay, let's see. Hold on, y'all. Maybe. Oh, there we go. Okay, let's see. Why view? Let's just do 125. No, golly. This is so sad. Okay, there we go. All right. And I'll make it big again. So which of the following correctly describes a feature of how enzymes function as reaction catalysts? So I'll give y'all 2 minutes. Tell me how they function as reaction catalysts. Oh, did I put it in poll? Because I want you to be able to pick more than one. So short answer. Sorry, y'all. Because it's choose one or more. Now you have to really analyze each one. You say one or more. It's tough. So I'm still going to give you the 2 minutes. Okay, 10 seconds if you haven't clicked in. Okay, so let's look at each of our answer choices, and then we'll grade our answers, okay? All right, so let's look at A. Enzymes function primarily by lowering the activation energy of a reaction in order to speed up the rate of a reaction. That is the definition of an enzyme, so check, that one's good. And y'all know what the activation energy is on an energy diagram? You've seen that before, like if you were, I want to do the other way, let me do the other way, right? If you start off with your energy here, that's your energy, and that's time going this way. This is your reactants, that's your products, right? And you know it does like this, and so what does your enzyme do? Does that. So it decreases your activation energy right there. Okay. All right, so most enzymes contain multiple protein subunits. Most proteins contain multiple protein subunits. This one's true. This one's true. And most enzymes are also usually part of almost like an assembly line. So they kind of like convert and then shuffle it off to the next one. Who converts to the next substrate, shuffles it off to the next one. And so it's very rapid, the way that they're organized to make metabolic functions go very, very quickly.
Is there any specific, like, because I took it as if there's multiple subunits, enzymes can do multiple things.
Well, a lot of times they do multiple things, like an ATPase, right?
But
Will
like
pump.
in like amylase versus like something like in the pancreas or.
Yeah, but usually even if it's amylase, even if it's different enzymes in the pancreas, usually those enzymes are going to contain multiple subunits. That they're gonna have, like, this subunit is going to be the catalytic subunit. This subunit is going to be the structural subunit, right? They're typically.
Not specific to like.
No, that is a very, very general, true statement. Very general. All right, C, adding the suffix ASE to the end of the protein's name denotes that protein is an enzyme. That is true. That is true. We learned that in the very first section, right? D, some of the amino acids in an enzyme's active site play a direct role in lowering the activation energy of a given reaction. This one's true. This one's true. So some of those amino acids, those are the ones inside of the active site, right? So those active site side chains are sticking out and they actually interact with the reactant molecules in order to help that reaction go. So it's specific protein-protein interactions, right? Because enzymes are super specific to their substrates, right? When you look at those enzyme names like alcohol dehydrogenase, right? There are specific things that enzyme will work on. And why is that? Because of those side chain interactions at the active site with the side chains of the target reactant molecule. It's all about those protein-protein interactions. So I was kind of tricky on that one, wasn't I?
I took it as like the enzyme was lowering reactions.
I didn't know you meant you wanted us to do all of them.
Well, I want to select, what did it say? Select one or more. A, B, C, D. Good. A, B, C, D. A, B, C, D. All right. But y'all at least chose more than one, so that's really good. Okay. Y'all ready?
We're gonna talk a little more.
Let's see. Oh, here we go. So it was all of them, right? I circled all of them. All right. So choose the statement that is false about genomic caretaker proteins. So we're looking for the false statement. It's still in short answer. Oh, this going back and forth between multiple choice and short answer. I'm sorry. Thank you, Taylor. I'm going to shoot for 1.5 minutes to cut it off. 1.5 minutes. Wow, I think we've had 40 in a while, actually. Ooh, 41. Getting excited. 5 seconds. I know sometimes technology can be the issue. All right. So enzymes that are required for DNA replication, repair, and recombination, are they one class? Yes, they are one class. So that is a true statement. That is a true statement. Proteins involved in like polymerase, you know, all those kind of things. Proteins involved in regulation of genes. So what is, that's true, it is. What's an example of something that regulates gene expression? Transcription. A transcription factor in general. There are many. There are many, many, many. Have y'all taken cell biology?
Yes.
Some, I think some people have.
I have.
Yeah. So all those kind of transcription factors that can either suppress or promote gene expression. Genomic caretaker proteins ensure that the integrity of genomic DNA is maintained and the expression of certain genes is controlled appropriately. Absolutely. What's an example of proteins that are involved in that? Ooh, that's a check mark. Somebody on the chat, what kind of proteins are involved in maintaining our genomic DNA and expressing certain genes that we've already talked about this year? That are non-specific DNA binders. Y'all are making me a little bit sad. Histones, right? They control how compacted or how loose kind of the DNA structure is, right? So we can regulate that, and we can regulate our histones. All right, heme-based proteins whose primary function is to either transport or store molecular oxygen are a type of genomic caretaker proteins. Nope,
That's usually described a transport protein.
that is a transport protein. That is correct. Okay, so what do we say? Y'all should have said D. Y'all said D. Good. Okay. All right, so I'm going to end it there. And so we got to click question 3. So we'll pick up on there tomorrow, Monday. And Monday I will have some more long answer thinking questions for you. But I don't have any for you today. Like I did before, print them? Yep, I'm going to print them just like I did before. So absolutely. And if you come by and you want a hard copy, I have extras in my office. So, okay. Any questions? Everybody good on Zoom? My internet wasn't working, so I missed attendance. Oh, that's okay. I'll see that you checked in. Tristan, would you send me an email real quick and just ask me to go in and modify your attendance? Because I can go in and do that. That's easy. Okay. Anybody else on Zoom need anything? Y'all good? All right, y'all send me, William, you can send me one too. Okay. All right, if nobody has anything else, I'm going to go ahead and end so that the recording will end. Okay, y'all have a great weekend.